All right, so here we go. We're going to talk about dihybrid crosses. I'm going to show you how to set it up. Guys, it's not difficult. It's just how you segregate the alleles that gets difficult. Now, just like a monohybrid cross, you're testing one trait for two different parents. Now with a dihybrid, you're going to be testing, because di means two, so now you're testing two traits for two different organisms or people or plants or whatever. So remember, dihybrid crosses testing two traits. Instead of having 4 squares, it's going to have 16 squares. So 16 squares. Again, pretty quick. You can do it. Setting it up is the most difficult. And there's another rule that I got to add so you don't mess it up. Okay, so a cross between two parents looking at two traits. Now what you're going to notice here is that I have something set up here. So like Mendel, he messed with peas, and so some of the traits that he looked at was whether the seed color was yellow or green, and so you have it here. And I even listed, I went ahead and I gave it a letter for its gene. So Y would be dominant for a green seed. So remember, if anywhere you see an uppercase dominant Y, that's going to be automatic green seeded, and then lowercase yellow, that's going to be a yellow seed. And then up here's your representations of it. And then another trait that he could have tested, that he did test, was seed shape. And if you look at the picture, you're going to notice that the seeds are either smooth and round or wrinkled. And so here you have green, this one's green smooth, green wrinkled, and here you have yellow wrinkled. So that's the kind of difference, so you can actually, in your mind when you're doing this, kind of picture what I'm trying to get across. And then if we assign it some letters, we're just going to say the capital R is going to be the dominant trait for having a round seed, and then the lowercase r is going to be the recessive for a wrinkled. So remember, anytime you see a capital R that's involved in there, it's probably going to automatically be a round, smooth looking seed. Now, here comes what I would consider the hard part. Okay? Now you notice I have this pea plant, so this could be a parent pea plant, and we're going to cross it with another pea plant. They are identical. Okay? So they're all heterozygous. Remember, heterozygous means they're different genes for each trait. So uppercase Y, lowercase, so dominant, recessive. Then with this parent, same thing. This is heterozygous for that yellow trait or that color trait, and then it's going to be heterozygous for that seed shape trait. And so here's, if I can make it and fit that in. So there's the dihybrid. Not difficult. Not difficult. Like I said, the hardest part is going to be taking it, because remember we said they were all heterozygous for their traits. So here's the hardest part. It's called segregating the alleles. Remember, law of segregation says one parent gives one gene. And so here's how we make sure the parent gives one gene. It's just like the FOIL method in math. Watch what I do. I'm going to start with this uppercase dominant Y. And it can only be paired with, over there, an R. We're looking for it to pair with an R. And so in this case, the capital Y is going to go with the capital R. So there's one square done. Now, if you notice, this dominant Y could go with this little r here. So you could just pair it, and that would be capital Y, lowercase r. So you can probably already see where this is going. It's so easy. You guys will be ahead of just the average college freshman biology taker. Okay, so that Y can't go with this uppercase Y, can't go with anything else, so it's done. So this one's left. So the lowercase is going to go with the capital R, and then it hasn't been paired yet with that one, so this lowercase Y is going to go with the lowercase r. Just like that. And since I've already told you that the other parent is identical, and all they're going to do is they're going to come over. It's going to be the same. We don't have to do it again. Okay? And so there, it's already set up. Now it just has to be solved. And if you remember your rules, remember the dominant comes before the recessive. Dominant before the recessive. But another rule is you keep like genes together. So the R's stay together and the Y's stay together. So whatever you see in the parents, that's the one that's listed first. In this case, it was the Y. And remember, Y's stay together and then R's stay together. So if I solve this quick, and I'll show you how to do it. And so you got a capital Y, capital Y, capital R, capital R. Remember, the parents were listed Y's first, so we have to list Y's first. And since they're all capital and dominant, it doesn't matter. Okay, and now we're going to go this one. So we have a capital Y, capital R. There's that rule. Okay, the dominant R goes before the lowercase. Okay, and you notice I'm not mixing them like this. I don't mix them up like that. They keep them all there. It's very simple to see. Okay, so I'm just going to shut up here and I'm going to solve so you can see how to do it. Okay, capital Y, capital Y, R, 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 Y, Y, R, R, 1, 1, R, R. See if you can beat me to get it done. Okay, there's my lowercase, and notice that's here because I have the Y up here that's dominant and it comes down first. R's with R's and Y's with Y's. There's a strange individual. Another one. Y, Y, R, R, Y, Y, little r, lowercase, lowercase. Okay, lowercase, lowercase. Okay. Okay. So if you take a look at it, that's what you should have. Now, for phenotypic ratio, guys, we could go through it. I mean, you can go ahead and you can figure out the colors and the seed shape, but I think what you're going to find is, if I've done this right, phenotypic. So what do they look like? Are they yellow or green? Are they wrinkled or smooth? You should be able to go through it, and you should have a phenotypic ratio. Phenotypic ratio. It should be 9:3:3 to 1. 9:3:3 to 1. So, go through it, see if you can figure out quick. Like, for example, you could go through and say, yellow. Okay, so we know that this is a yellow, and we know it is, and if I can't remember exactly what I had there. Oh, it was green. Green, and round is the dominant. Okay. So if I set that up again, this is going to be green, round. And so you're going to go through and look and see how many green round there are. There should be 9 green round ones. And then go through and list it. You can make sure I haven't messed up and made any mistakes. So pause it, pause the video here, and go through and see if you get a 9:3:3 to 1. Okay, so one thing I kind of want to get to before, we had the law of segregation, right? Mendel had a law of segregation where one parent gave one gene. Now, he also came up with another one that you kind of need to know. It's a test that's going to be on the test, and it's called the law of independent assortment. Okay, so we're getting ready to talk about how sperm cells and egg cells are made and how the genes are shuffled inside. And the law of independent assortment just states that the genes, whenever a sperm cell or egg cell is made, they're sorted at random. It's like shuffling cards. It's kind of hard to shuffle cards and do it a planned way. Now, I'm sure there may be some people could do it, But if you, just the average person, just sit there and shuffle cards, you won't be able to tell that you're putting the ace here, here. It could go anywhere. And so our genes are sorted at random through the formation of gametes. And so a gamete is just a sex cell. That's another term, but really it's just a sperm cell or an egg cell. Okay, so a gamete is a sex cell. Sex cells can either be that, sperm cell or egg cell. And the cool thing about this law here, I showed you a picture of like the Jonas Brothers, the picture of me, myself and my son Eli when we were in kindergarten, and some of the other actors. Even though we look the same, you know, we're similar, just because of this law of ended assortment, it ensures that we are genetically different. So like my son and I, we are genetically different, even though we share some things. In the Jonas Brothers, you could definitely see some of the things that they share, which they are genetically different. So if you look really close, they are they're not identical twins. So law of independent assortment said everything's sorted at random. It's like a random shuffling of the cards, and it ensures that we all are different.